Share my screen. I'm a little distracted. I was eating lunch right before logging in and I spilled oil all over my clothes. I was hoping to have time to change and I ran out of time. So I'll try not to stand up so you can see the oil all over my clothes. All right, today we're continuing, whoops, a little too far, on chapter 1, and that includes a little bit of chapter 10. It includes pages 264 to 272 of the current version of the textbook in chapter 10, classification of microorganisms. In the lab today, we'll be doing lab 01. Let me remind you, you need to turn in the worksheet for lab 00 by Friday at 8 a.m. or else I will, well, I'll boot you from the class because I'm taking that worksheet for attendance. Any questions about any of that? All right, if there's no questions, let's begin. Let's see. Right here. Hopefully I marked this. Yes, we talked about that. All right, we were talking about we find microorganisms essentially everywhere. And this slide is addressing where is everywhere on the planet Earth. Environments where microorganisms are found include on the land and in the water, in the atmosphere. And yes, you can find microorganisms up about a mile in the air. You can find microorganisms subsurface on the land and under the ocean floor. And also you can find microorganisms down about a mile from the surface of either the land or the ocean floor. You find microorganisms on plants and animals, and we'll talk a little bit more about that later. All plants and animals have a community of microorganisms living with the plant and animal. And we call that the microbiota. Essentially, wherever liquid water is present, you can find microorganisms. The temperature needs to be between a negative 20 degrees Celsius and 120 degrees Celsius to find microorganisms. Below a minus 20 degrees Celsius, you can find microorganisms, but they'll be frozen and not metabolically active. Microorganisms, some of them, especially bacteria, can survive freezing temperatures, but they will not be metabolically active below minus 20 degrees. Above 120 degrees Celsius, it's hard to find microorganisms because this elevated temperature begins killing microorganisms. Any questions about any of that? All right. Microbes are numerically the most abundant life forms on the planet Earth. How many microorganisms are there? And we're just talking about prokaryotic cells. If you include viruses, the numbers would be 10 times higher than the numbers I'm going to be talking about here. How many microorganisms do you find in a gram of soil? A gram of soil, if you don't know, is, I don't know if you can see my fingers, but about that much. You actually find about 10 to the 8th bacteria and archaebacteria and fungi and other cellular microbes in a gram of soil. And that will depend on the soil conditions. Generally, the wetter the soil, the more microorganisms you'll have, and the more you have organic matter in the soil. What am I trying to say? Decomposing matter in the soil. You'll have more microbes. Any question about any of that? All right. So in a millimeter of ocean water, and that's about 1 gram of ocean water, do you have more or less microbes than 10 to the 8th per milliliter? Anyone want to guess? Come on, it's not a hard guess. It's either yes or no. You got a 50-50 shot at the very worst.
I'll guess more.
Anyone else?
Less.
Less. Yeah, actually, ocean water is considered one of the deserts of the planet Earth. And so there are less microbes in the ocean water than in a gram of soil. And that's just mainly because there's not a whole lot of nutrients in ocean water. Anything else I want to say about that? Nah, that's it. In a gram of human feces, do you find more or less than 10 to the 8th microbes per gram?
More.
Yes. Generally, you find more. Once again, this depends on how fresh the human feces is and how dry it is. Like if somebody were to go to the bathroom in the desert and then you would see it, I don't know, a week or two later and it's all crispy, there would probably be less because the water has evaporated and the bacteria have died. But generally, especially if it's fresh, there will be more than that. On one human being, come on, that one's easy. More. The entire Earth, of course, there's more. Let's talk about some of the numbers. Where do we have water? I think I'm hiding it here. Not water. What do we have? Soil, there it is. So in 1 gram of soil, assuming it's average moisture like Western Oregon or Western Washington, which actually that's probably more than average moisture, and then average soil decomposition, meaning you got stuff for the microbes to live on, about 10 to the 8th. It can be less, like 10 to the 6th, especially in the desert. Ocean water is only about 1 times 10 to the 6 microorganisms in a gram or a milliliter of ocean water. And the ocean is a desert on the planet Earth. Human feces, generally, if it's a fresh feces, it's around 1 times 10 to the 11th microorganisms per 1 gram of feces. And actually 55% of the weight of human feces is from the weight of microbes. That's just something to store as trivia. Of course, if it was a very dry human feces, like I stated, somebody goes to the bathroom in the desert and you take a look at it a week or 2 later, it'll be much less. And actually, there would only be about 1 times 10 to the 6th, which is about the same microorganisms you find in the desert soil or in the desert ocean water. Any question about any of that?
I have a quick question.
Yeah.
So is this going over information that's going to be pertinent to our lab 1? Just so I make sure I'm looking at like all the right documents while we're going over it.
This information is not going to be pertinent really to lab 1. It will tell you that, you know, you can find more microbes in certain places than in others. And that part's pertinent. But the exact numbers, I'm not going to test you on these. And it's not really pertinent to lab 1.
Okay,
This
thank you.
one, This one is of interest, and that is, how many microbes do you find on your average human? It's 3.8 times 10 to the 13th microorganisms, which are cells. And that's an astounding number of microbes on each person. Now, obviously, this is your average person. A child is smaller, for example, so they'll have a little bit less, but it'll be around that number. On the entire Earth, if anyone is curious, there is around 1 nonillion. That's 1 times 10 to the 30th, or 1 followed by 30 zeros after it. Okay, you probably never even heard of a nonillion before. And the human population is between 7 billion and 8 billion, just to give you a comparison. And that is, there's many, many more microbes on the planet than there are humans. Any question about any of that? Okay, microorganisms can cause disease. We'll be talking about that a little bit later in the term. A microbe that causes disease, we call it a pathogenic microbe. That is one capable of causing disease. Most microorganisms do not cause disease, so you should know that most microorganisms do not cause disease. Only a small percentage of microorganisms are pathogenic, meaning disease causing. Any question about any of that? Now this you can expect on the quizzes or exams. When we're talking about pathogenic microbes, it's important to talk about infectious versus inherited diseases. An infectious disease is a disease caused by our microorganisms. And it's infectious because it can spread from one individual to another. What's an example of an infectious disease? Come on, somebody's got to know an infectious disease. They're all around us.
AIDS.
AIDS is an infectious disease. HIV is the infectious agent. What else? Let's go at least with.
Would a cold be considered an infectious?
Yes, a cold is an infectious disease. It's caused by a microbe. It's a virus, and it can spread from one individual to another. So it is an infectious disease. Okay, it's a little different in the sense that it's caused by a virus, and they're a little different microbe because they don't have cells. But other than that, it is an infectious disease. All right, an inherited disease is caused by a person's genetics. So they have the wrong genes, and then they get the inherited disease. What is an example of an inherited disease?
Celiac disease.
Celiac disease. What's another one? I didn't hear you. Somebody gave an answer, but I didn't hear it. Can you get a little closer to your computer and repeat that? Well, can somebody else give an inherited disease? Let me turn my speaker up.
Hemophiliac disease.
Yes, hemophilia. And that's caused by somebody having 2 defective genes, or if you're have the correct hemophilia, there's more than one. And it's on the X chromosome, in which case boys only would have 1 defective gene because they only have 1 X chromosome.
Is blindness is it inherited?
I'm not aware of anyone getting defective genes and then getting blindness, but certainly it's possible. And if you have an environmental cause of blindness, like somebody who, I don't know, was always looking at the sun and then became blind. Galileo was somebody who that happened to. That would not be an inherited disease because he got that from looking at the sun. But if you're born with it, meaning somebody's born blind, well, yeah, that's probably because of defective diseases, genes, in which case it's an inherited disease. I'm not aware of blindness running in families, though, so that's why I'm questioning. I'm not sure that blindness is an inherited disease, but it might be. If you have the wrong genes and then you're blind with birth, you would be defective or an inherited disease. But I just don't know an example of that. If somebody knows somebody who was born blind and it's a genetic thing, you can mention it and then they'll say, oh, there's an example. All right, any questions? Let's move on. We're looking at some microorganisms here, and they're small. Let me blow this up a little. Slideshow. There's actually a scale down here, so that's 1 micrometer, also called 1 micron, which is the size of your average bacteria. And the width actually there is a little bit less than 1 micron. This is a little bigger. Can somebody read that? I can't read it. Blow it up even more. Yeah, 15 microns right here. And this one is bigger than 50 microns. And then there's 50 microns again. And actually this is one organism. It's a colonial microbe. And these right here are baby colonies inside the mother colony. And you see each of these dots? Let me blow that up a little. Right there, there's a dot. That's one cell. One cell, and they're connected together to make a circular colony. And then these are baby colonies inside. This will open up, and then the babies will come out and go elsewhere and colonize someplace. That looks like a white blood cell, and this is about 10 micrometers. Also well, not exactly a microbe, but a microorganism because we can't see it with our naked eye. And then there's some fungi there. Nope, I take that back. That's a lymphocyte, like a T lymphocyte with HIV budding off of it. So that would be a microorganism there, a virus. You can tell that microorganisms come in various shapes and sizes. That's all I'm trying to state here. Oops, that was wrong. And the slideshow. Microorganisms play beneficial roles for us and in the environment, meaning for the planet Earth. We talked about them, that microorganisms are important in decomposing organic waste. When a tree dies in the forest, it's generally the microbes that'll decompose it. It is true there are some fungi, like mushrooms, which are not microorganisms, and they can also decompose decaying organisms, but most of it is done by microbes. Microbes are also important in being producers in the ecosystem. They can be photosynthetic producers or chemosynthetic producers. The photosynthetic producers are important because they convert sunlight into glucose, the energy of glucose, and they convert sunlight and carbon dioxide and water into glucose. And then they produce oxygen. The chemosynthetic organisms are similar, it's just they use chemicals to make their own food. Any question about any of that? We mentioned, excuse me, I got a frog in my throat. We mentioned that microbes are important in the nitrogen cycle. The nitrogen cycle runs on the planet Earth because of microbes converting nitrogen from one part in the ecosystem to another part, from one form to another form. Microorganisms are also used to produce things like industrial chemicals, ethanol that you can put in your car and run with it. They add it to the gasoline. The production of acetone, microbes can make that. We use microorganisms to make some insecticides. And these tend to be less harmful for the environment than man-made insecticides. We use microorganisms to make fermented food. That includes vinegar, cheese, bread, beer, chocolate. We use microorganisms to make some products in manufacturing, like cellulase. We use microorganisms to make some antibiotics, like penicillin G. And then there's many other products that we use that are produced by microorganisms, like some used in medicine, like insulin. We make almost all insulin now in microorganisms. They cloned out the human insulin gene and put it in a microbe to make the insulin. And that they did a long time ago. My father was a diabetic and got insulin. And I'm thinking he started getting human insulin like in the 1970s, certainly in the 1980s. All right. Biotechnology and genetic engineering is our next topic. Biotechnology is the use of microbes and other organisms to produce foods and products for human use. So biotechnology is centuries old. An example of biotechnology is people have been breeding corn for centuries. It started with the American Indians, and they bred corn to make Indian corn. If you don't know what Indian corn is, it's a little cob, sort of like this can here, a little smaller than my face and multicolored, kind of pretty. And then scientists took those corn, and I think Westerners did too, and they made a yellow and white corn, and then they bred that. And so now they have corn cobs where you can get the corn or the kernel of corn, 80% of it is oil in it. And they use these corn for making corn oil. And I assume they bred corn for making protein as well and other things like that. Any question about that? Anyways, that's biotechnology, centuries old, just using microbes and other organisms to produce foods and products for human use. And then you say, well, I thought biotechnology was using, I don't know, modern technology in a lab to make things. That's actually genetic engineering, a new technique of biotechnology. Applications of genetic engineering include producing a variety of proteins from bacteria and fungi, such as making vaccines and enzymes in a microbe, or we could say, well, that's genetically modified bacteria, where you put a human gene or another gene into the bacteria to make the bacteria produce a product for us. And that we've done many times, but the first time it was done was actually to make the human insulin. We do genetic modified bacteria to protect crops from insects, coat the crop with the bacteria, or to coat the crop with the bacteria and then help the crop from freezing. So genetically modified bacteria. Gene therapy, another technique of genetic engineering. With gene therapy, somebody is born with a defective gene, and then you use genetic engineering to put the correct gene in the person. The best known examples, and this has been going on for 20 or 30 years now, is children who are born with a defective immune system, and they have defective genes, and so their immune system isn't functioning properly. You can take out some of their immune cells, grow them up in vitro, and then add the correct genes to those cells, and then put those cells back in the child. And then the child will now have a functioning immune system. It works well with the immune system because you don't need to add that many cells to the child to get it to be corrected. If somebody were to be born with defective skin, like an albino person, you have to correct all of the skin cells, or at least most of the skin cells. And with the immune system, you only need to correct a few cells, and then the child will have a functioning immune system. All right, any questions about biotechnology or genetic engineering? If not, let's switch gears again and talk about how microbes are important in human welfare. Humans are a microbial ecosystem. Wherever you look on the planet Earth, you practically find a microbial ecosystem, but humans have a bunch of microbes with them, so that when we look at a human, they have a microbial ecosystem with them. Microbes are important in ecology. We already mentioned that microbes, bacteria and archaea, are important for running the nitrogen cycle. Well, microbes are also important for recycling carbon in the Earth's ecosystem. And you kind of know this when a tree dies, that carbon isn't locked up in that dead tree forever. Microbes will dissolve the tree, recycle the carbon, and then make the carbon available for other organisms. And that's true for the nutrients of the tree. So microbes are important for recycling nutrients on the planet. They're important for recycling sulfur on the planet and phosphorus on the planet. And the sulfur system and the phosphorus system run much like the nitrogen cycle. It's just that the sulfur and the phosphorus cycles, or we'll call cycles, they have more steps where microbes are not involved in the step. Whereas in the nitrogen cycle, almost all of the steps involve a microorganism. But there are a few that do not. And the sulfur cycle, like if sulfur is in a rock, you can get weathering of the rock by wind and air, and then the sulfur will be dissolved by the water and converted by an organism, largely microbes, into different forms. But the point is that the sulfur came out of the rock by means other than microbes. Anyways, these are some examples of the microbial mediated biochemical cycles that support life on Earth. And that's the carbon cycle, the nitrogen cycle, the nutrient cycle, the sulfur cycle, and the phosphorus cycle. These cycles were first studied by Winogradsky and Bajorink from the 1880s and the 1892s. And they were the first to show that microorganisms were important in running these cycles. Any question about any of that? Winogradsky was actually unique, and that is he was a scientist who did not study microorganisms in their pure form to study the microorganism. Winogradsky studied microbes together in a community and then looked to see what the microorganisms in their community did. And he needed to do that in order to understand the microbial mediated biochemical cycles. And that's probably why he was the first. Like I said, most microbiologists study an organism when it's been purified and it's the only microbe to study. And to study the nitrogen cycle, for example, you got to study many different microbes because one microbe runs one step, but the next step is ran by another microbe. Any question about any of that? All right, microorganisms are also important for humans and the planet in that we can use microorganisms for bioremediation. Bioremediation means life and to remedy. So when there's a spill or an environmental problem, we can use living organisms to help deal with the environmental problem. As you know, we use microbes to help deal with breaking down sewage. We discussed that. Well, you can use bacteria and other microbes to break down other pollutants. Like in this oil spill, they're spraying microbes on the oil so that the microbes can degrade the oil and then get rid of the pollution. You can also use microbes on other pollutants like mercury. Now, the microbe does not degrade the mercury, but it can convert it to a less toxic form. Okay, so bioremediation, where you use microbes to degrade organic matter and degrade or detoxify pollutants, making them less harmful. Any question about any of that? Do any of you guys remember the Exxon Valdez accident, which happened in Alaska, where they had a humongous oil spill? It was the largest oil spill to hit the United States. And one of the ways they treated that was bioremediation, putting microbes on the oil to help digest it. Nobody's going to comment on that? All right, let's move on. Microbes can be useful in that we can use them as biological insecticides. There are some microbes that are pathogenic to insects, and they can be an alternative to man-made chemical pesticides. Generally, the biological insecticides is not as toxic or as harmful to the environment as the chemical pesticides. And they can be as successful in reducing crop damage and disease transmission as the chemical pesticides. For example, Bacillus thuringiensis is a bacteria which is fatal to many insects. And there we're looking at Bacillus thuringiensis right here. And yet Bacillus thuringiensis is harmless to humans and other animals, as well as harmless to plants. And there is some Bacillus thuringiensis you can buy, and then you can spread it on your crop. My father, way back in the 1960s, bought Bacillus thuringiensis to apply to apples instead of using a chemical pesticide. The trouble was, my father was a little lazy, and he did apply it once or twice, but the Bacillus thuringiensis has to be applied to the apples about the same as the chemical pesticides. And the chemical pesticides, if you don't know, when you're treating apples, you need to treat the apples every 2 weeks once the apples start to form, all the way until you pick the apples. Although you don't want to spray it what do you say, during the week before you pick the apples. So you should not spray during that time. Well, the Bacillus thuringiensis has to be applied every 2 weeks. And on top of that, after it rains, and we were in Western Oregon, which is like Western Washington, and it rains a lot. And you can imagine when it rains, the Bacillus thuringiensis is washed off the apples. So after the rain, you need to apply it again. And my father was much too lazy to do that. So the apples did have worms, but there were less worms from his 1 or 2 applications. Any question about any of that? As I stated, it would have worked if he would have applied it every 2 weeks and apply it also after the rain, which in the fall in Oregon, Western Oregon, it rained a fair amount. All right, any questions about microbes and how they're useful to humans or important in the Earth's ecosystem? Any question about that? If not, we're going to move on and talk about the naming and classifying of microorganisms. That was started by this scientist here, starting the naming and the classifying of microorganisms. Does anybody remember who that was from your prerequisite biology class? We did discuss this person. Who is it? He did start the naming of organisms as well as he started the classifying of organisms.
Carolus Linnaeus?
Yes, Carolus Linnaeus is the Latinized name, and he actually was Swedish, and I think his name was Carl Linné, but he liked Latin so much he Latinized it, and most people call him Carolus Linnaeus. Your book calls him Carl Linnaeus, meaning they're using the Latinized last name, but I guess the Swedish first name. But he's the one who started the naming and the classifying of microorganisms. And he did that in 1735. In the naming of organisms, he made it so that each organism has 2 names, their genus name and the specific epithet name, meaning the species specific epithet. But you can't say it's the species name because sapiens is not our species name. Homo sapiens is humans' species name. And Homo is the genus, sapiens is the specific epithet for the species. Any question about that? We call this naming system the scientific nomenclature, and it's also called the binomial nomenclature because each name, each species name, has 2 words in the name, and that's what binomial means, 2. Any question about any of that? All right, so we're going to talk a little bit more about the naming of things. But remember that Carolus Linnaeus also started the classification of all organisms. He was the first person who grouped all species into different groups. And he tended to put everything into the kingdom Plantae and the kingdom Animalia, meaning animals. And nowadays we have more than just 2 kingdoms. We'll talk about that later. But he also did the scientific naming system, and that has continued from his day. I mean, we're still using his naming system. Let's move it here. The binomial scientific nomenclature system has all names as either italicized or underlined. Generally, when you're typing, you will italicize the scientific name. But if I were to go up on the board and then write it, like Homo sapien, you're supposed to underline it, because I can't write in with italics. When we're giving the binomial name, the genus name, meaning the first one, is capitalized, and the specific epithet is always lowercase. So Homo sapien, we mentioned, capital, lowercase. And this is italic or underlined. The names are Latinized and they're used worldwide. What we mean by that is Homo sapien is used everywhere in every language to describe the species humans. So it's a worldwide naming system. Everybody in the world uses it. And if you use Homo sapien, everyone who knows that will know that you're referring to humans. The name can be descriptive or honor a scientist. Homo sapien is descriptive. A homo means walks on two legs. Sapien means wise. Homo also means man, walks on two legs. So Homo sapien means literally wise man. Escherichia coli, which most people call E. coli, is both descriptive. Coli is where it's found in the colon. Escherichia is named after Esther, an early microbiologist. So it is honoring that scientist. Staphylococcus aureus is descriptive. Aureus means golden. And that's because Staphylococcus aureus in color are yellow golden. And then Staphylococcus is descriptive. Coccus means it looks like a ball. Staphylo means it is balls. Staphylococcus mean balls that take on the shape of a grape cluster. Any question about any of that? Once you use the scientific name for the first time, all other times you can abbreviate the name. You abbreviate the first letter of the genus and then write out the specific epithet, like Staphylococcus aureus and Escherichia coli are found in the human body. And so the first time you spell it out, and then all other times you can abbreviate it. We're going S period aureus. It's found on the skin and the nasal passageways, perhaps in the throat too. And E. coli is found in the intestines and the colon. E. coli is one that is so common that even among scientists, we frequently will call it E. coli the first time, and everybody knows what we mean. But generally, you're supposed to say Escherichia coli the first time, and then E. coli all other times. But like I stated, E. coli is so well known that even among scientists, they will go E. coli. Any question about any of that? All right, so we've talked about the naming of microorganisms. Now let's talk about the classifying of microorganisms. Before we can address the classification of organisms, we first need to look at microorganisms. We need to look at the classification of all living organisms on the planet Earth. We classify all organisms into a 3-domain system. So all organisms are classified into 3 domains, and we can call these 3 domains the tree of life, because here's the tree of life, and it sort of looks like a tree with 3, I don't know, 3 trunks maybe. Any question about that? Why we classify all living organisms into 3 domains is we classify organisms based on genetic data, meaning all of the bacteria are more similar to each other genetically than an organism in any other domain. So we can sequence an unknown organism and then determine its DNA sequence and then compare that sequence to other organisms. And the domain will be the organisms that are most similar to that organism, meaning the most similar in their sequence of DNA. So the domain system is defined, and it's based on the genetic data, the sequence of the DNA in that organism. This came as a shock to biologists because we thought the bacteria and the archaea or the archaea bacteria were similar to each other before they sequenced the DNA, because they're both prokaryotes, they're both single cells, they're both simpler compared to a eukaryote. But then when we sequenced the DNA, we found the archaea have a totally different sequence of DNA than the bacteria. And they are as different from each other as they are to the eukaryotes. In fact, if you look at this very closely, let me see if I can blow this up. Yeah, but it's smaller, not larger. Here we go, kind of. If you look at this very closely, lost it again. The archaea and the eukarya have a more similar line from right here to there. They're actually the same line. And that's because they're a little bit more similar in their DNA, just a little hair similar in their DNA than the archaea are to the bacteria. So the archaea are more similar to the eukaryotes, just a little, than they are to the bacteria. Any question about any of that? I think I accidentally advanced that. There we go. Go back to that slide. So there are 3 domains: the bacteria, the archaea, also called the archaea bacteria, and the eukaryotes, or the eukarya. Those are the 3 domains. Now, each of these domains has a kingdom, and we're going to use the 6 kingdom approach. So the bacteria, this domain only has 1 kingdom, which is the bacteria. The archaea also only have 1 kingdom, which is the archaea. But the eukarya have 4 kingdoms. They have the kingdom Plantae, the plants, and Animalia, the animals, the fungi, the fungus, and then the kingdom Protista, the protists. Any question about any of that?
What domain did you say the plants, animals, fungus, and the other one? What domain are they a part of?
Showing it right here. They're in the domain eukarya.
Oh, I see. Thank you.
Okay. And what did I want to say about that? Oh, there is no definition for what is a kingdom. So people can argue about, this animal or this organism should belong to this kingdom. And another scientist can argue, no, it belongs in this kingdom. And there are arguments about that because there is no what do you call it, definition for a kingdom. In fact, when we looked at the kingdom Protista, it is defined as a eukarya. That's not a plant, not an animal, not a fungi. So this is a collection of other organisms which are eukaryotes, which are not plants, animals, and fungi. And it's important to realize the Protista do not have a monophyletic origin, meaning they may not be similar to each other. Let me point this out here. All the fungi came from a common ancestor. All the animals came from a common ancestor. All the plants came from a common ancestor. All of these others did not, unless you come way back here, come from a common ancestor. In which case, way back here, all the eukaryotes came from a common ancestor. That's what we mean. The Protista are a collection. They are not necessarily similar to each other. And we just use that as a grab bag to put other eukaryotes into that are not plants, animals, and fungi. Because of that, scientists actually argue about how many kingdoms there are. Some argue there's only 4, is that right? Some argue there's 5. Some argue there's 6, and that's the approach we'll use. The bacteria, the archaea, and then the plants, animals, fungi, and Protista. And probably the majority use the 6 kingdom approach. But others say there's more, and you can divide the Protista very easily. So you can break it down into 9, 10, or 20 different kingdoms, largely just by dividing the Protista. You can also start dividing the plants, and then the archaea and the bacteria into different kingdoms, and then you can argue there's 20, 30, or 50 different kingdoms. And scientists haven't agreed on how many kingdoms there are, and they've been arguing about that for, I think it's been over 20 years now. So I don't think they'll ever decide how many kingdoms there are. And the problem is that nobody agrees because there is no definition for what a kingdom is. So we'll just use the 6 kingdom approach, which I've got shown here. And that is there's a bacteria, that's a kingdom and a domain, archaea, which is a kingdom and a domain, and then kingdom plants, animals, fungi, and Protista. Any question about any of that? All right, let's move on.
Sorry, I have a really quick question. I'm just looking at the lecture worksheet
Uh-huh.
and it's asking what domain we will mostly be looking at in this class. Which one would that be?
We'll mostly be looking at the domain bacteria.
Okay, thank you.
In the 3-domain system, it's important to realize that they have different cell types. The bacteria have prokaryotic cells, so the cell type is prokaryotes. The archaea also have prokaryotic cells, so their cell type is prokaryotes. The eukarya have eukaryotic cells, so their cell type is eukaryotes. Okay? Your textbook calls this a cell type. You don't need to memorize this for an exam, but some people call this a super kingdom. Super because it was before and a higher than kingdoms. And the super kingdoms were around before the domains were around. Otherwise, we would have called it super domain. And so you could, in the classification, first divide things into a super kingdom, meaning you divide into prokaryotes and eukaryotes. And then you divide the eukaryotes, which isn't divided, into a domain. And then you divide the super kingdoms into the archaea, and the super kingdom prokaryotes into the archaea and the bacteria. Okay, so just know that there are cell types or super kingdoms, and that there are two, the prokaryotes and the eukaryotes. Any question about any of that? All right, if not, let's talk about some of the characteristics about the domain bacteria. As I just stated, we'll talk about the domain bacteria a little bit more in this class than any other domain. And there are some reasons for that, and we'll discuss that later. The cell type is they're prokaryotes. The bacteria have the molecule of peptidoglycan in their cell wall. So if the bacteria has a cell wall, you'll find peptidoglycan in the cell wall. And this is a molecule which is the main molecule in the cell wall. The bacteria multiply mostly by binary fission, and that is the cell will grow and then literally divide into two, split into two. That's what binary fission means. For energy use, the bacteria can get their energy needs from all of the sources. They can get their energy from an organic chemical, and that's what we do. We get our needs from glucose and other food. That's an organic chemical. And E. coli does that. Some bacteria can get their energy needs from an inorganic chemical, and that can be sulfur or nitrate or hydrogen sulfide or iron. And generally, the bacteria that get their energy needs from an inorganic chemical is usually a chemosynthetic organism. And we'll talk a little bit more about that later in the term. And then some of the domain bacteria get their energy needs from photosynthesis, meaning they get their energy needs from light and then convert that energy of light into chemical energy and make sugar out of it. And those are the photosynthetic organisms. You should know that most of the domain bacteria are free living species, free living decomposers, they decompose things, or free living primary producers, like the photosynthetic organisms or the chemosynthetic organisms, primary producers. But some of the bacteria are parasitic, meaning they live on and off of a host and do damage to that host. And some of the bacteria are pathogenic, meaning they're bacteria which cause human disease. The main reason why we're mostly going to study the bacteria in this class is because the bacteria cause human disease. And the focus of this class is on microbes that cause human disease. Any question about any of that? All right, let's move.
I
to
do have a question about,
Go ahead.
sorry. I did have a question about number 9 on the lab or on the lecture sheet. Just want to double check. It says E. coli and a few other names are so widely known and used to the general public. It is more often used than blank, which is not correct. Is that the scientific name?
Yeah, the scientific name.
Okay, I just want to make sure I got that right. 'Cause
Now
I
you
don't
got to
think
give the
that's incorrect.
scientific name. Okay.
Yes.
Yeah. All right.
Perfect.
Just say the scientific name. You have to give the scientific
The
name
actual
for E.
name.
coli. Yes. Yes.
Perfect. Thank you.
Okay. Some characteristics of the archaea. They are cells that have prokaryotic cells. Does anybody know, do the archaea have peptidoglycan in their cell walls? Nobody's
Yes.
going to hazard? Somebody said yes. Anyone else?
No.
Generally, the archaea do not have peptidoglycan in their cell walls. There are a few exceptions, and we're not going to talk about them in this class. And they only have a little bit of peptidoglycan. But generally, the archaea lack peptidoglycan. So peptidoglycan is a molecule generally only found in the bacteria. Some of the archaea are found in extreme environments, and these include the methanogens, archaea that produce methane, the extreme halophiles. Halophile means salt lover. So you can go like in the Dead Sea between Jordan and Palestine, and you'll find halophiles living in the Dead Sea. Most of them are archaea. You can find archaea as extreme thermophiles. That means they live in very hot environments, like the boiling mud baths of Yellowstone National Park or in the geysers. But when we've looked further, and this has been known now for at least 20 years, we find the archaea everywhere. It is true that some of them are found in extreme environments, but most of them are found all over the planet Earth. And so you can find the archaea everywhere. In fact, when we look at the bacteria in the soil and in, oh gosh, what do you call it? The bacteria in the, not the bacteria, the microbes in the soil and in the ocean, the, what do you call that? The plankton. The archaea are a significant portion of those microbes in the soil and in the plankton. Okay, any question about that? So wherever we look on the planet, we can find the archaea. The earlier versions of our textbook kind of had that wrong. It used to say that the archaea were mostly found in extreme environments. That isn't true. They are found everywhere on the planet. It is true that in extreme environments, you can find the archaea. And generally, you don't find too many other organisms. But you can find some bacteria in all of these environments. But the archaea tend to dominate. But the archaea are found everywhere on the planet. And does anybody know, are any of the archaea pathogenic? Do the archaea cause human disease? Does anyone know? Nobody knows? So I'm going to answer. There is no known archaea that causes human disease. And that's the main reason why after this lecture, I'm not going to be talking very much about the archaea. They do not cause human disease. And in a sense, that's a good thing. So if we work with the archaea, it doesn't cause us disease. In a sense, if you believe in evolution, sooner or later, the archaea will evolve to growing on or in people and then causing disease. And that will be a problem when that happens, because we really haven't developed many drugs to fight the archaea, because there are no archaea that currently cause human disease. Any question about any of that? All right, if there's no questions about the archaea, let's turn to the characteristics of the eukarya. There are 4 kingdoms in the eukarya: the plants, the animals, the fungi, the protista. All of these kingdoms and all of the eukarya lack peptidoglycan. None of these cells have peptidoglycan in their cell wall. And of course, the animals don't have a cell wall. Some of the protista don't have a cell wall as well. The kingdom fungi, singular fungus. You don't really need to know. They have cell walls that are a little different, made of chitin. You don't need to know that. They use organic chemicals for energy. Mainly the fungus degrade something to get their energy needs. So most of them are decomposers. There are a few yeast, which are pathogens causing disease in humans. And I think you know that from a yeast infection, human pathogen. There are multicellular molds and mushrooms, which consist of mycelia, which have filaments of hypha. And there we're seeing a hypha right there. But many of the fungi are unicellular, and that includes some of the molds and all of the yeast. And the yeast shown right here, they're all unicellular. All right, any questions about the fungi?
It's 6:10.
Yeah, I got 609. Let me finish with the No, I did finish with the fungi. Never mind. So let's put it right here. And I will see you in the lab. Any questions?
Do we have access to this PowerPoint other than your lecture?
You will after I'm done with the lesson.
Perfect. Thank you.
All right. All right, I'll see you in the lab at 6:20.